Hi everyone, this is a session on classification of carbohydrates and simple carbohydrates. There's a separate session on complex carbohydrates. And I do understand that you're all apprehensive about learning core biochemistry, right? So no worries. My assurance to you is that this is not a complete core factual biochemistry session, because in the first half of the session, I will be telling you a few facts like what are carbohydrates, what are the various types of carbohydrates, and what is a simple carbohydrate, what are the various types of simple carbohydrates and examples. But using those facts as a foundation, in the next half of the session, I will be helping you to understand few clinical conditions like lactose intolerance. So this is going to be a clinical biochemistry session. With that assurance, let's start the discussion. Okay. Now, the first fact that we are going to discuss about is what are carbohydrates? I feel instead of me telling you the definition of carbohydrates and you just memorizing the definition, it will be easier for you to remember if you try to derive the definition of the same by looking at these two structures. So I've shown you structure of glucose on one hand and structure of fructose on the other hand. Okay, the good news is you don't have to memorize any of these structures. Just observe these two structures and let me know one basic similarity between the two, one difference between the two. From wherever you are, you can answer to yourself what is strikingly similar between glucose and fructose. Don't you see that both glucose and fructose have multiple hydroxyl groups? Yeah, so this is the similarity. And what is the difference? Do you see that glucose has got an aldehyde group as a functional group, whereas fructose has got a ketone group as a functional group? So what is the similarity? All carbohydrates have multiple hydroxyl groups. The difference is glucose has got an aldehyde group, whereas fructose has got a ketone group. This is why we define every carbohydrate as polyhydroxyaldoses or ketoses. Isn't that a logical definition? Yeah, all carbohydrates are polyhydroxyaldoses or ketoses having the general molecular formula CnH2nON, or on hydrolysis they give rise to molecules of molecular formula CnH2nON. So this is the definition of carbohydrates. Now what are the types of carbohydrates? You classify carbohydrates in the first level into two types. What are the two types? The first one is simple carbohydrate. The next type is complex carbohydrate. The name tells you everything. What do you think is a simple carbohydrate? A simple carbohydrate has got only carbohydrate units in them. Whereas complex carbohydrates, in addition to carbohydrate units, they have either lipids or proteins attached. So what are complex carbohydrates? In addition to carbohydrate units, they have either lipids or proteins attached. In that case, how can you classify complex carbohydrates? If a carbohydrate is attached to a protein, you can call it as a glycoprotein or as a proteoglycan. So what are the first two types of complex carbohydrates? Glycoproteins and proteoglycans. I want you all to understand that these two terms are not synonymous. Glycoproteins and proteoglycans are totally different. They are two different types of complex carbohydrates. If a carbohydrate is attached to a lipid, you can call it as a glycolipid. So the third type of complex carbohydrate is glycolipids. So what are the three types of complex carbohydrates? Glycoproteins, proteoglycans, and glycolipids. And we have a separate session on complex carbohydrates. What are we going to learn about now? We are going to learn about simple carbohydrates. So how do you think you can classify simple carbohydrates which have only carbohydrate units in them? Logically, the classification is possible only based on the number of carbohydrate units they have. Isn't that logical? Based on the number of carbohydrate units they have, we are going to classify simple carbohydrates into three types. The first one is a monosaccharide. The next type is an oligosaccharide, and the third type is polysaccharides. What do you think is a monosaccharide? Monosaccharides have only one carbohydrate unit in them. Whereas oligosaccharides have from 2 till 10 units, which means disaccharide also comes under this classification. Disaccharide is an example of oligosaccharide which has got 2 units. So what is a polysaccharide? If a simple carbohydrate has got 11 or more number of units, then that is called as a polysaccharide. So that's about classification of simple carbohydrates. First, about monosaccharides. Interestingly, monosaccharides which have only one carbohydrate unit in them can be classified in two ways. The first way is depending upon the number of carbon atoms they have. Second way of classifying them is based on the functional group that is present. Now, depending upon the number of carbon atoms they have, you can classify monosaccharides as trioses. The first type is trioses, they have tetroses, pentoses, hexoses, and so on. So, what are the types of monosaccharide based on the number of carbon atoms they have? If they have 3 carbon atoms, you will call them as trioses. 4 carbon atoms, you will call them as tetroses. 5 carbon atoms, pentoses. 6 carbon atoms, hexoses, and so on. Now, how do you classify monosaccharides based on the functional group that is present? Simple. If a monosaccharide has got an aldehyde group, you will call it as an aldose. If it has got a ketone group, you will call it as a ketose. So these are the two ways of classifying monosaccharides. If you find it difficult to understand this classification of monosaccharides, follow me by looking at these two structures again. What are the two structures? Again, glucose and fructose. For both glucose and fructose, I'm going to ask you 3 questions. If you answer these 3 questions, you will be able to understand the classification of monosaccharides. First question is, in glucose, how many functional groups do you find? I've already told you a carbohydrate will have either an aldehyde group or a ketone group as the functional group. That way, how many functional groups do you find in glucose? In glucose, there is only one functional group, which means it is a monosaccharide. So, your glucose is a monosaccharide. And what is that functional group which is present? It is an aldehyde group, which means your glucose is an aldose. How many carbon atoms do you find in glucose? Don't you see that glucose has got 6 carbon atoms, which means your glucose is a hexose. So, based on the functional group that is present, your glucose is an aldose. Based on the number of carbon atoms present, your glucose is a hexose. Now, the same 3 questions for fructose as well. How many functional groups do you find in fructose? Only one functional group, which means your fructose has got only one sugar unit. So, fructose is a monosaccharide. Do you understand this? Fructose is a monosaccharide. And what is that functional group which is present? It has got a ketone group, which means your fructose is a ketose. How many carbon atoms do you find in fructose? There are 6 carbon atoms. So, your fructose is a hexose. So, do you understand the difference between glucose and fructose? Both glucose and fructose are monosaccharides. Both glucose and fructose are hexoses. The only difference is glucose is an aldose and fructose is a ketose. So, glucose and fructose are functional isomers because both share the same molecular formula C6H12O6. That's all about classification of monosaccharides. And in this classification of monosaccharides, I would like to tell you about 2 facts which can be asked as MCQs. The first fact is based on a misnomer. What is a triose? I told you. I said triose is a monosaccharide with 3 carbon atoms, right? Triose is a monosaccharide with 3 carbon atoms. The misnomer is maltotriose. So, maltotriose is not a triose, it's actually a trisaccharide with 3 glucose residues. When I say maltotriose has got 3 glucose residues, how many carbon atoms do you expect? 3 into 6 is 18 carbon atoms. So, don't you think it's a misnomer? Triose is supposed to have only 3 carbon atoms. Maltotriose is not a triose, it's a trisaccharide with 3 glucose residues. Suppose they ask you, all of the following are trioses except. It can be a simple MCQ. All of the following are trioses except. And if one of the choices happens to be maltotriose, go for it because maltotriose is not a triose, it's a trisaccharide. And the other fact that I want you all to understand at this point of time is, whenever you see the term malt in a compound's name, if you see the term malt in a compound's name, it means it's made up of multiple glucose residues. For example, maltose is made up of 2 glucose residues. Isomaltose is also made up of 2 glucose residues. Maltotriose is made up of 3 glucose residues. So malt means multiple glucose residues. So that's one fact regarding classification of monosaccharides. The next fact about classification of monosaccharides is, how do you name a ketose from the corresponding aldose name? How do you name a ketose from the corresponding aldose name? I will give you a clue. Erythrose is an aldose, erythrulose is a ketose. Ribose is an aldose, ribulose is a ketose. Pseudoheptose is an aldose, pseudoheptulose is a ketose. So how do you name a ketose? By adding UL to the corresponding aldose name, you will name a ketose. Why will you have to know this? Suppose they ask you an MCQ. All of the following are aldoses except. The question is, all of the following are aldoses except. One of the choices happens to be something with UL. Example, ribulose or pseudoheptulose. You now know it is not an aldose, it's a ketose. Okay, so that's all about classification of monosaccharides. Now let's start discussing about disaccharides. What are disaccharides? I said it's one of the types of oligosaccharides. They have 2 sugar units. And as far as disaccharides are concerned, you will have to know 2 facts about the commonly asked 5 disaccharides. So what are the 2 facts that you need to know? The first fact is, what are the individual sugars that are present in a disaccharide? Second fact is, you will have to know what is the linkage that is present. For example, if I ask you about sucrose, you should be able to tell me that sucrose is made up of glucose and fructose. Don't you all know that? Sucrose or the table sugar which we consume is nothing but a disaccharide with glucose and fructose. That's not enough. You should also be able to tell me that the linkage that is present in sucrose is alpha 1,2. So what are the 2 facts that you will have to know about all the 5 disaccharides? The individual sugars that are present. First fact is, you should know the individual sugars which are present. Second fact is, you will have to know the linkage that is present. Okay, so first let's learn about individual sugars, then let's learn about the linkage that is present. Okay, so first about maltose. Maltose, isomaltose and trehalose. What did I tell you about malt? Malt means multiple glucose residues. So maltose, isomaltose, trehalose, all 3 are made up of 2 glucose residues and they differ only with respect to the linkage that is present. So now we know about maltose, isomaltose, trehalose, all 3 being made up of 2 glucose residues. They differ only with respect to the linkage that is present. The 4th disaccharide, just now I told you about, it is sucrose. So what is sucrose made up of? Sucrose is made up of glucose and fructose. So the 4th disaccharide is sucrose, which is made up of glucose and fructose. And the last disaccharide is lactose, the one which is present in milk. Lactose has got galactose and glucose. Don't change the sequence. Don't say it is glucose and galactose. Sequence matters here. For you to call something as lactose, the first sugar has to be galactose and the next one has to be glucose. So that's all about the individual sugars which are present. Let's now try to learn about linkages. And as far as linkages are concerned, instead of me telling you about linkages which are present in every disaccharide, it's easier for you to remember if I tell you the basic thumb rule based on which every carbohydrate prefers to get linked. And then if I tell you the exceptions, it will be easier for you to remember and recollect during the exam. So let me tell you the thumb rule first. The basic thumb rule is all carbohydrates prefer to have alpha 1,4 linkages along straight chains. All carbohydrates prefer to have alpha 1,4 linkages along straight chains, and at branch points alone they prefer to have alpha 1,6 linkages. Yeah, so I'm going to show you the same as an image. Have a look at this. Do you see that all these carbohydrates along straight chains are linked with alpha 1,4 linkages? Wherever a branch point begins like this, you find only alpha 1,6 linkage. At the origin of branch point, you find alpha 1,6 linkages. Thereafter, it is going to be sequentially alpha 1,4 linkages. So this is the basic thumb rule. Along straight chains, alpha 1,4 linkages. At branch points, alpha 1,6 linkages. Okay, have this in your mind. Now about maltose, isomaltose and trihalose. Maltose follows the basic thumb rule. So what is the linkage that is present in maltose? The basic thumb rule is they prefer to have alpha 1,4 linkages. So maltose has got alpha 1,4 linkage. How about isomaltose? S for 6. So isomaltose has got alpha 1,6 linkage. Trihalose has got alpha 1,1 linkage. So what is the linkage present in maltose? Alpha 1,4, isomaltose alpha 1,6 and trihalose is alpha 1,1. Now about sucrose. I will tell you a fact about sucrose. Though sucrose is made up of two reducing sugars, glucose and fructose, sucrose as such is non-reducing. Do you understand this? Though sucrose is made up of two reducing sugars, glucose and fructose, sucrose as such is non-reducing. My first question to you is, why do you say glucose and fructose are reducing? We call glucose and fructose as reducing sugars because both in glucose and fructose, there are free carbonyl groups. Any carbohydrate which has got a free carbonyl group will be reducing. And that is why glucose, which has got a free carbonyl group in the first carbon atom, and fructose, which has got a free carbonyl group in the second carbon atom, are reducing sugars. But why is sucrose non-reducing? Because we get sucrose by linking the first carbon atom of glucose with the second carbon atom of fructose. Do you understand this? Why is sucrose non-reducing? Because we get sucrose by linking the first carbon atom of glucose with the second carbon atom of fructose. That way, neither the carbonyl group of glucose nor that of fructose is free. So this tells you that the linkage that is present in sucrose is alpha 1,2. What is the linkage that is present in sucrose? You are linking the first carbon atom with second carbon atom. So what is the linkage? It is alpha 1,2. Okay. So fill up this tabular column. Sucrose has got what linkage? Sucrose has got alpha 1,2 linkage. That is why it is non-reducing. Now about lactose. What is the linkage that is present in lactose? For this, you will have to know one basic fact. Do you all know that human cannot digest cellulose? Yeah, human cannot digest cellulose. That is why plant diet comes under fiber diet. Because if you cannot digest something, the undigested food will reach the colon, will act as a fiber, will attract water. So why do we say plant diet acts as a source of fiber? Because plant has got cellulose and cellulose cannot be digested by human enzymes. And the reason behind this is human digestive enzymes. Most of the human digestive enzymes cannot attack beta linkages. This indirectly tells you that the linkage that is present in cellulose is, yeah, cellulose. What is cellulose made up of? Cellulose is made up of multiple glucose residues. Yeah, cellulose made up of multiple glucose residues linked by what linkages because of which we cannot digest it? Cellulose has got multiple beta 1,4 linkages. Okay. And if I tell you that the only human digestive enzyme which can attack beta linkage is lactase. The only human digestive enzyme which can attack beta linkage is lactase. What does that tell you about lactose? Lactose has got beta linkages. Yeah, lactose has got beta linkage and it is beta 1,4. So can we quickly recollect all that we have discussed about disaccharides? I told you about 5 disaccharides. First, we discussed about the individual sugars which are present and then we discussed about the linkages. So maltose, isomaltose, trihalose, all 3 are made up of 2 glucose residues and they differ only with respect to the linkage that is present. Sucrose, the table sugar, is made up of glucose and fructose. Lactose, the one which is present in milk, has got galactose and glucose. That's about individual sugars. Linkages, maltose follows the thumb rule. So what is the linkage that is present in maltose? It is alpha 1,4. Isomaltose, S for 6, it has got alpha 1,6 linkage. Trehalose has got alpha 1,1 linkage. Okay. Sucrose. I said sucrose is non-reducing because you get sucrose by linking the first carbon atom of glucose with the second carbon atom of fructose. So what is the linkage that is present in sucrose? It is alpha 1,2. Now lactase is the only human digestive enzyme which can attack beta linkage. So lactose has got beta 1,4 linkage. Okay, so that's all about disaccharides. Very simple, right? Now having known about disaccharides, let's try to understand about a clinical condition called as lactose intolerance. So you must have learnt about so many other disorders, right? For example, fructose intolerance or galactosemia. So the basic difference between lactose intolerance and other disorders is lactose intolerance is a digestion defect. Yeah, what is lactose intolerance? A defect of digestion. Whereas fructose intolerance, galactosemia, all these are metabolic defects. Why am I calling it as a digestion defect? Because lactose intolerance is caused by the defect of lactase in the intestinal villi. What will happen when lactase is absent in the intestinal villi? Any lactose that is present in the diet cannot be digested to give galactose and glucose. If you're not able to break a disaccharide into monosaccharides, disaccharides cannot be absorbed. Only monosaccharides can be absorbed. So this disaccharide will stay back in the lumen. And because lactose, the disaccharide is osmotically active, it attracts water. And this causes osmotic diarrhea. So can you tell me what is the predominant clinical presentation of lactose intolerance? Lactose intolerance always presents with osmotic diarrhea because it is caused by lactase deficiency in the intestinal villi, in which case lactose in the diet cannot be digested. When it is not digested, it stays back in the lumen, attracting water, causing osmotic diarrhea. And when something is not getting digested in the intestinal lumen, undigested substances will reach colon. Now, what is expected in colon? Your colon is loaded with microorganisms. And all these microorganisms start utilizing lactose. And they convert lactose into hydrogen or methane. All intestinal microbes, they act on lactose which is not digested to give rise to hydrogen and methane. And both hydrogen and methane are gases. And because gases accumulate in the colon, what are the other manifestations of lactose intolerance? It presents with bloating. It presents with flatulence. It presents with frothy stools. So bloating, flatulence and frothy stools, all these can be explained by the production of hydrogen and methane in the colon by intestinal microorganisms. And the same hydrogen and methane generation is also the explanation for hydrogen breath test and methane breath test done to detect lactose intolerance. So if the question is, what is the investigation of choice for lactose intolerance? What will be your answer? Hydrogen breath test and methane breath test. How is this test done? You will ask the person to fast overnight, and the early morning breath sample is taken, and in the breath either hydrogen is measured or methane is measured. It is laboratory's preference. So you measure either hydrogen or methane in the early morning breath sample. And then a measured quantity of lactose is given to the individual, and then at periodic intervals we start collecting breath sample and we keep estimating hydrogen or methane concentration in the breath sample collected periodically. And if you see that hydrogen or methane concentration keeps increasing beyond physiological limits, then it is diagnostic of lactose intolerance. So hydrogen breath test and methane breath test are done to detect lactose intolerance. Additionally, colonic microorganisms act on this undigested lactose to form acids. And these acids are responsible for acidic pH of stools. So in lactose intolerance, what is the pH that you observe in stool? It is acidic pH. And this acidic pH is responsible for perianal excoriation. Yeah, that is responsible for perianal excoriation, which is one of the features of lactose intolerance. So quick recap of lactose intolerance. The first fact is it's a digestion defect, not a metabolic defect, because it is caused by the defect of lactase in the intestinal villi. When lactase is absent, undigested lactose stays back in the lumen, attracting water, causing osmotic diarrhea. Undigested lactose reaches colon. In colon, microorganisms utilize lactose and convert them into hydrogen and methane, both of which are gases. That is responsible for bloating, flatulence, and frothy stools. And generation of hydrogen and methane is the basis of hydrogen breath test and methane breath test done to detect lactose intolerance. Microorganisms also convert lactose into acid. That is responsible for acidic pH of stools, and that is responsible for perianal excoriation. Okay, so that's about lactose intolerance. So don't you think biochemistry is interesting? If somebody tells you that biochemistry is a preclinical subject, please don't agree to that. Please don't accept that. Yeah, because if you don't understand biochemistry, you will not be able to understand any of the clinical conditions. For every clinical condition, there is always a biochemical basis. So all our sessions are going to be clinical biochemistry session. Okay. So that's all about lactose intolerance. I've explained you all this. Okay, and that's about monosaccharides, that's about oligosaccharides. Let's now learn about polysaccharides. What are polysaccharides? I told you. Polysaccharides are carbohydrates which have 11 or more number of units. Right? Now, how do you classify polysaccharides? These polysaccharides can be classified into two types. Yeah, one is homopolysaccharide, the other one is heteropolysaccharides. And as the name indicates, in homopolysaccharides, individual units are the same. They are made up of repetitive units of one sugar moiety. Whereas in heteropolysaccharides, the individual units are different. And the best example for heteropolysaccharide is mucopolysaccharides, otherwise called as glycosaminoglycans. So please memorize it, which is the best example for heteropolysaccharides. It is mucopolysaccharides, otherwise called as glycosaminoglycans, about which we'll be discussing in the session on complex carbohydrates. For now, what are we going to discuss about? We are going to discuss about homopolysaccharides. These homopolysaccharides, based on the function they perform, can be classified into two types. The first type is a structural homopolysaccharide. The second type is storage homopolysaccharide. And as the name indicates, as far as physiology and biochemistry are concerned, you don't have to hunt for the meaning anywhere else. The meaning will be there in the term. What do you think is a structural homopolysaccharide? It acts as a structural component of a cell. Whereas storage homopolysaccharides will act as storage forms of carbohydrates. In that case, between structural and storage homopolysaccharides, which do you think will be branched? Storage homopolysaccharides will be branched, because only when it is branched, you can accommodate more number of molecules in a limited space. So all storage homopolysaccharides are branched, structural homopolysaccharides are unbranched. Okay. Now, what are the examples of structural homopolysaccharides? I have three examples for structural homopolysaccharides. We have cellulose, about which I told you just before. Apart from cellulose, it is inulin. Yeah, cellulose, inulin and chitin. So, what are the three examples of structural homopolysaccharides? Cellulose, inulin and chitin. So first about cellulose, we have discussed everything about cellulose. I told you cellulose is a component of plant cell wall, and this cellulose is made up of multiple glucose residues. Yeah, linked by what linkages, because of which we cannot digest it? They are all linked by beta-1,4 linkages. So cellulose is a homopolysaccharide made up of glucose present in plant cell wall. It is linked by beta-1,4 linkages. Now about inulin, the one which we use for measuring glomerular filtration rate. Yeah, and this inulin is present in plant tubers. and it is a homopolysaccharide made up of fructose. This was asked once in NEET PG. Inulin is a homopolysaccharide made up of fructose. Now about chitin. Chitin is present in, where have you heard of chitin? The moment you hear the term chitin, what comes into your mind? It's a part of exoskeleton. Yeah, chitin is a part of exoskeleton of crustaceans. For example, scales of snakes. Yeah, scales of snakes or scales of cockroaches. All these are made up of chitin. Now, what is chitin made up of? Chitin is a homopolysaccharide made up of N-acetyl glucosamine. Chitin is a homopolysaccharide made up of N-acetyl glucosamine. If you're asking me, how do I remember it? It's easy. Didn't I just now tell you that chitin has got something to do with snakes? Yeah, chitin has got something to do with snakes. Snake is nag. What is nag? N-acetyl glucosamine. So chitin is a homopolysaccharide made up of N-acetyl glucosamine. So these three are the examples of structural homopolysaccharides. Let's now learn about storage homopolysaccharides. So what are the examples of storage homopolysaccharides? Easy to remember, right? Which is the storage form of glucose in plants? In plants, glucose is stored as starch. Which is the storage form of glucose in animals? In animals, it is glycogen. So what are the two examples of storage homopolysaccharides? Starch in plants and glycogen in animals. So let's learn about the structure of starch first and then about structure of glycogen. So first about starch. The moment you hear the word starch, doesn't porridge come into your mind? Yeah, porridge. Yeah, we have porridge for breakfast, right? So starch and porridge, they resemble each other in multiple ways, right? Starch, porridge comes into your mind. And don't you all know that porridge always has got two components? One is a solid component. The other one is a liquid component. It is because starch has got two structures within itself. One is highly branched amylopectin. The other one is unbranched amylose. So what are the two structures present within starch? One is highly branched amylopectin. The other one is unbranched amylose. When I say amylopectin is branched, what does that tell you about its structure? It means amylopectin will have multiple glucose residues. Yeah, amylopectin will have multiple glucose residues which are linked by what linkages along straight chains? Along straight chains, you will find alpha 1,4 linkages. At branch points alone, you find alpha 1,6 linkages. Thereafter, it is going to be sequentially alpha 1,4 linkages. So this is the structure of amylopectin. So amylopectin will have multiple glucose residues linked by alpha 1,4 linkages along straight chains. At branch point alone, there is alpha 1,6 linkage. Thereafter, it is sequentially alpha 1,4 linkages. And because amylopectin is branched, it is very stable. And because it's stable, its melting temperature is high, say 50 degree. If its melting temperature is high, at room temperature, it is yet to melt. So that accounts for the solid part of starch. So what is the reason for solid part of starch? It is amylopectin which is branched. Now tell me about amylose which is unbranched. Easy, right? Amylose will have multiple glucose residues linked by what linkages? Linked by just alpha 1,4 linkages along straight chains. Because they don't have branches, they are not that stable. Because they are not that stable, their melting temperature is less. And because their melting temperature is low, at room temperature, they've already melted. And that will account for the liquid part of starch. So starch, what are the two components? Branched amylopectin and unbranched amylose. That's all about starch, storage form of glucose in plants. Now storage form of glucose in animals is glycogen. Please listen to this carefully because you understanding the structure of glycogen is essential for you to understand glycogen metabolism. And only when you understand glycogen metabolism, you will understand all glycogen storage disorders. Okay, so glycogen. What are the two tissues wherein glycogen is stored in human body? In human body, glycogen is stored in liver and in muscle. So the first fact is glycogen is stored in liver and in muscle. Second fact, glycogen is a highly branched structure ever known. Suppose the MCQ is, which is the most branched carbohydrate? What will be your answer? Your answer should be glycogen. Okay. Third fact is, it's a spherical molecule. Your glycogen is a spherical molecule. And in the center of the sphere, you find a protein called as glycogenin. In the center of the sphere, you find a protein called as glycogenin, to which every glucose residue is attached directly or indirectly. Suppose it's a glucose residue which is attached in straight chain, it is directly attached to glycogenin. If it's a glucose which is there in branch point, again it is indirectly attached to glycogenin. So in the center, you find a protein called as glycogenin, to which all the glucose residues are attached directly or indirectly. And every straight chain has 11 to 13 glucose residues. So 3, 6, 9. Every straight chain has got 11 to 13 glucose residues linked by what linkages? Linked by alpha 1,4 linkages. At branch points alone, you find alpha 1,6 linkages. And the entire structure is arranged in 12 concentric layers. I said every straight chain has got 11 to 13 glucose residues. It's 12, right? So every glucose residue in a straight chain will form one concentric layer. So entire glycogen is arranged in 12 concentric layer. And in such a structure, where will you find more number of branch points? Will you find more number of branch points towards the center or towards the periphery? Let me answer that for you. You find more number of branch points towards the center. As you move towards the periphery, the number of branch point decreases. Logically, because you start synthesizing glycogen from the center and you proceed towards the periphery. When you start the synthesis, you have more number of glucose molecules. So you'll introduce as many branch points as possible. And as you move towards the periphery, you don't have many glucose molecules free in hand. So you stop creating branch points. So you find more number of branch points towards the center and the number of branch point decreases as you move towards the periphery. So that's all about glycogen. Glycogen is stored in liver and in muscle. It's the most branched structure ever known. It's a spherical molecule. In the center of the sphere, you find a protein called as glycogenin, to which every glucose residue is attached directly or indirectly. And every straight chain has 11 to 13 glucose residues linked by what linkages? Alpha 1,4 linkages. And at branch points, you find alpha 1,6 linkages. The entire structure is arranged in 12 concentric layers to enable compactness. And in such a structure, you find more number of branch points towards the periphery. No, you find more number of branch points towards the center, and the number of branch point decreases as you move towards the periphery. Okay, so that's all about the facts and concepts related to this topic. Now it's time that we learn about, or we try to understand the same concept using few MCQs, one-liners. Okay, let's start with one-liners. The first one-liner for you is glucose and fructose are examples for what? They are functional isomers because glucose is an aldose and fructose is a ketose. Second one, inulin is a homopolysaccharide made up of what? Inulin is a homopolysaccharide made up of fructose. Third one, chitin. Yeah, it is present in scales of snakes. What is snake? Nag. So it's a homopolysaccharide made up of N-acetyl glucosamine. Okay. The next one. Non-reducing disaccharides are. I already told you about one non-reducing disaccharide, which is sucrose. Didn't I tell you? Yeah, one is sucrose because the linkage is alpha 1,2. The other one is trihallose. Why is trihallose also non-reducing? Because trihallose has got 2 glucose residues. And what is the linkage that links the 2 glucose residues? I told you alpha 1,1. Where do you find the free carbonyl group in both the glucose residues? In the first carbon atom. So both the carbonyl groups are not free. So not only sucrose, even trihallose is non-reducing. Please memorize it. Sucrose and trihallose are 2 non-reducing disaccharides. In starch, which is unbranched and which is branched? Many a time students get confused here. Please remember amylose. Yeah, amylose is unbranched and amylopectin is branched. Okay. Let's discuss few MCQs. This is the first MCQ. All of the following are trioses except. I gave you the same MCQ when we discussed also. Yeah, all of the following are trioses except. What is your right answer? It is maltotriose. Because maltotriose is a misnomer, it's not a triose, it's a trisaccharide with 3 glucose residues. Next question. The linkage that is present in lactose is. How did I ask you to remember this? Lactase is the only human digestive enzyme which can attack beta linkage. So lactose has got what linkage? Beta 1,4. The linkage present in isomaltose, S for 6. Isomaltose has got alpha 1,6 linkage. All of the following are aldoses except. Which is not an aldose here? Glucose definitely is an aldose. Galactose is an aldose. Ribose is an aldose. What did I tell you about ribulose? If you find UL, it means it is a ketose. Now, few clinical case-based MCQs. A 21-year-old man presents with diarrhea, bloating, flatulence and frothy stools every time he consumes milk, ghee and butter. The probable enzyme defect in this condition is. Isn't that a very simple question? Following intake of diet which is rich in lactose, the person is presenting with diarrhea, bloating, flatulence and frothy stools. So what is your diagnosis? Your diagnosis is lactose intolerance caused by the deficiency of lactase. Okay. Now the same scenario. Yeah, similar scenario. A neonate presents with diarrhea, incessant cry and frothy stools. Perianal rashes are prominent. The neonate responded well when the child was started on rice porridge. Rice does not have lactose. So after withdrawal of lactose from the diet, the child has responded. So we know for sure it is lactose intolerance. But what is the investigation that you're going to ask for in this child? What is the investigation I told you for lactose intolerance? For lactose intolerance, I said hydrogen breath test and methane breath test. But can you order for hydrogen breath test and methane breath test in a neonate? Can you ask the neonate to fast overnight? Can you be taking periodically breath samples in a neonate? That's going to be very difficult. So if it's a neonate, all that you can ask for is stool pH. I said stool acidity is one of the features of lactose intolerance. So if it's a neonate, it is stool acidity. Okay. And for exclusion's sake, for your benefit, let me tell you about other choices in a brief way. Schilling's test is done to detect B12 malabsorption. Please memorize it. Schilling's test is done to detect the presence of B12 malabsorption. Guthrie's test is a screening test done to detect phenylketonuria. Methane breath test can be done in adults, not in neonates for lactose intolerance. Okay. Now, one image-based MCQ. This is the last MCQ for this session. Identify the structure. So, you are shown 2 sugars here which are linked by a linkage. How can you identify the structure? The first fact is, in both the sugars, see how many carbon atoms are included in the ring. If the ring includes 5 carbon atoms, it is glucose or something else. Okay. That is, if the ring includes only 4 carbon atoms, it is fructose. If the ring includes only 4 carbon atoms, it's fructose. Now look at this second sugar. In the second sugar, how many carbon atoms are included in the ring? 1, 2, 3, 4. Only 4 carbon atoms are included in the ring, which means this is fructose. Okay, now look at this. It is 1, 2, 3, 4, 5. 5 carbon atoms are included in the ring, which means it is either glucose or galactose. It can be glucose or galactose or mannose. So how will you find out whether it's glucose or galactose or mannose? Check the second and the fourth carbon atoms alone. Yeah, look at the second and the fourth carbon atom. In both the carbon atoms, if OH lie below the plane of the ring. In both the carbon atoms, if OH lie below the plane of the ring, it is glucose. So what is this? This is glucose plus fructose. Okay, glucose plus fructose. So what is it? That is sucrose. Okay, now you're looking at a ring. You're looking at a ring which includes 5 carbon atoms in the ring, and then you're checking at the first and the fourth carbon. You're checking the second and the fourth carbon atom. OH in the second carbon atom is above the plane of the ring. Instead of being below the plane of the ring, in the second carbon atom, if OH lies above the plane of the ring, that is mannose. In the second carbon atom, if OH lies above the plane of the ring, that is mannose. In the fourth carbon atom, if OH lies above the plane of the ring, that is galactose. Have you understood this? How will you find out which sugar it is? The first fact that you should check is how many carbon atoms are included in the ring. If the ring includes 4 carbon atoms, it is fructose. If the ring includes 5 carbon atoms, it is glucose or mannose or galactose. How will you find out whether it's glucose or mannose or galactose? Just check second and the fourth carbon atoms. If in both second and fourth carbon atoms OH lie below the plane of the ring, it is glucose. If in the second carbon atom OH lies above the plane of the ring, it is mannose. In the fourth carbon atom, if OH lies above the plane of the ring, it is galactose. In this case, it's a disaccharide made up of glucose and fructose. So it is sucrose. Okay, answer is sucrose. So that's all for this session. Meet you all in the next session, wherein I will be discussing about heteropolysaccharides and about complex carbohydrates. Thank you.